Oh, well, I don't have any other written questions coming in. So if it's okay, let's move to the panel discussion because I'm sure Richard and Philip have questions that they'd like to ask and we should get them on. Is that okay, Darryl?
Absolutely. Thank you.
Okay. That was a fantastic presentation. I really liked it. So.
In response to your question, Charlie, I think that, yeah, it would be really fun to go over the details of this experiment that Darryl is running to understand. It seemed like the soluble hypoxide hydrolase activity was increasing later in the day 4 to 6 range. So it does give you a little bit of a window potentially to get in there and therapeutically try to manipulate the lipidome and in a beneficial way, as Darryl eloquently, showed. But I agree, you probably are going to want to treat first and study mechanism later, given the potential severity here.
So what do you think about that mechanism for long haulers? Is there an opportunity for precision nutrition here? Okay, and I say that specifically so we could hear from Richard and Philip, experts in the area.
So, Richard or Philip?
Oh, sorry, Bruce. I thought you were going to go.
I wasn't sure.
No. It raises. I'm happy Bruce presented some of that data and updated us. Clinically, that they're seeing these effects up to 9 months. I think you said now, Bruce,
Yeah.
and it's a good question. I'll frame nutrition a little differently in that, and I think Philip will agree with me. Everybody should be eating well no matter what, right? So it's a little different than the pharmacological model that some of the other people are doing with. So, now's your chance to get your nutrition right and see what happens. So I think it poses an interesting question because I'm starting to become more and more convinced now over the past years that higher omega-3 intakes lead to increased blood levels, at least in humans, and animal models, tissue levels of some of the specialized pro-resolving mediators. Do they go up high enough? That's, I think, a big question we still have. I worry a lot in the elegant biochemistry of this, that some of the rate-limiting steps are going to be enzymatic in molecules with short half-lives. But I think there's an opportunity there, and there's definitely testable hypotheses and an important clinical question with these long haulers. What are we going to do there? And can we, can a small difference of nutrition over a long period of time add up enough to make a difference here? I think that's important.
Has anybody looked retrospectively at a cohort of long haulers to see whether there's a difference in severity or symptoms relative to their diet pre-COVID?
I haven't seen it yet.
I haven't seen it yet either.
No, I don't. Yeah, I can jump in if. Yeah, I don't think that's happened yet, but undoubtedly it will. But just to follow up on Richard's point, so I do agree that good nutrition is important and will give people a lot of the sort of metabolic wherewithal that they need. But it's very difficult for most nutrition to deliver the parent omega-3s that all of these wonderful compounds that we've been hearing about are derived from. So I think this sort of takes the omega-3 part of the story out of nutrition and into the gray area heading towards pharmacology, because I think the amounts of EPA and DHA that would be required to drive this process are more than most sort of diets can deliver. We published a study on oxylipins as a function of omega-3 supplementation and showed, dose-dependent increases in serum oxylipins with supplemental omega-3s. Of course, at taking people on a normal diet, you've got pretty low levels, but you can really drive them up with, you know, moderate to high level of supplementation. I guess there's variation between people, which is partly determined by their dietary intakes. But I do think we're getting beyond, Richard, I don't know if you do agree, we're getting a little bit beyond the realms of nutrition when we're talking about driving these mediators up by letting physiology do its work.
Yeah, I think we're on the same page, Phil. I think, you know, pharmacologically, you know, I think is the most promising approach for this. But we do see some differences, I think, in retrospective analyses on COVID outcomes by nutritional status.
Oh, yeah, sure, yeah.
And there might be a little bit of a window there, but I'd be careful. Even those retrospective analyses, we're talking about, you know, relative risk or odds ratio, of, you know, 20%ish. They're not too big. Of an effect. So
and also you don't know. I mean, you know, you can't really establish cause and effect, but you don't know whether any benefit that's seen is due to SPMs or due to other mechanisms that might be important.
Absolutely. You know, I think people eat well, exercise well, have better body weights, you know, and less diabetes risk. A lot of things that have shown up here in the epidemiology of COVID. I agree with you.
Well, the most recent studies that I've seen coming from the Tufts Nutrition Center on supplementation put the break point at about 2.5 grams. And then at that level, then you begin to see peripheral blood SPM levels go up.
Yeah.
And but as you point out, that doesn't tell you about the potential autocoid actions in target tissue.
That's right. Yeah.
But that's true for all lipid mediators. But there's some light in there, as you heard from Bruce Levy's presentation, this very clear impact of statins. And I wanted to ask Bruce, because Bruce has studied statins, and statins do have an impact on lipoxygen biosynthesis and through polyisoprenyl phosphates. So do you want to go into that, or nuanced a little bit too much here?
I'm happy to briefly. One other thing I would think is worth bringing up is the study that was published a few years ago in the New England Journal of Medicine, giving supplementation to a population of pregnant women and looking at the outcome of their offspring. And they found significant protection from wheezing, but also protection from respiratory infections. And so it's a clear example of supplementation, and it was dose dependent, providing protection against respiratory infections. So just one other supplementation trial to add to the mix. In terms of statins, there's many potential ways they could benefit. One is that we see that they can actually induce the formation of some SPMs, and this is actually in a related to the soluble epoxide hydrolase story, the cytochrome P450 story that Darryl mentioned. So there's an intersection there, interestingly, in the lipid metabolism for production of SPMs that's distinct from its pathway effects on HMG-CoA reductase inhibition. We have found that one of the products of mevalonate phosphorylation, presqualane diphosphate, is an polyisoprenyl diphosphate that appears to be an intracellular target for the SPMs to control neutrophil activation. And this is also relevant in macrophages. And it controls NADPH oxidase, phospholipase D, controls PI3 kinase in a very potent way. And we're working on that pathway actively in our research. HMG-CoA reductase inhibition clinically, of course, doesn't shut down the enzyme, it just modulates it. So you'd still see production of this downstream polyisoprenyl diphosphate. So we think that's an important intracellular mechanism for regulating neutrophil and macrophage function. And it's unknown at this point whether the EETs that Darryl talked about do regulate this molecule, but it's of interest.
Thanks, Bruce. I do have to mention A question came in for Dr. Zeldin from the audience that I'll read to you. Actually, two questions that are relevant. One is, how do you think that the SH epoxide hydrolase inhibitor is affecting prostanoid production? How did it affect 6-keto PGF2 alpha? Do you have some thoughts there, Darryl?
Yeah, so I don't think it's a direct effect. So I don't think we're dealing with off-target effects of the SEH on cyclooxygenase activity or the activity of one of the synthases. I think what we're probably dealing with is the effect of SEH overall on the inflammatory process within the lung. You know, the reduced pro-inflammatory icosanoid storm, the reduced cytokines that are then resulting in decreased COX-derived prostaglandins. So you're kind of shutting off that vicious inflammatory cycle.
Right.
So indirect effect is what we think is going on.
Do you think that the SH inhibitor has an impact on lipase pathways? Because you know, that would be an obvious target. Do you think that? Have you or Bruce looked at that?
We haven't looked at it. I think it's pretty selective. At the doses we use it, it's super selective for the soluble epoxide. In fact, it doesn't even cross-react. There are several epoxide hydrolases. There's a microsomal epoxide hydrolase. There's EPHX3, which is another epoxide hydrolase. And the inhibitors that the urea-based inhibitors that he's developed are highly selective for the soluble form. So I don't know that there would, at least I've not read any results of papers that have suggested that there's off-target effects on other pathways.
Okay, so there's another interesting question, general audience. This is from Michael Sussman. He's asking about whether the virus itself at all responsible for the production of inflammatory lipids. And yes, that's a very interesting question. Does anybody want to take it? No. The viruses would be a great place to start, and we've looked at some of the viral genomes. There doesn't appear to be any obvious cyclooxygenase or lipoxygenases there. But remember that every time a virus buds, in this case from an airway epithelial cell, it carries a little bit of the cytosol from the host. So it's possible that, and you know, the airway epithelial cells are rich in cyclooxygenases 1 and 2, the soluble epoxide hydrolases, lipoxygenase, depending on where you are along the airway, pinch off part of that cytosol and take with it an enzyme from the host. It's certainly possible, but hasn't been demonstrated. It's just something that comes in your dreams at night, right? I don't think there's any direct evidence. Although, Dipak, you pointed out a couple of papers where the lipid mediators have an action directly on the virus, some of the Japanese papers, and I think that that's pretty exciting. Do you want to expound on that?
Well, yeah, I mean, they had direct antiviral activity, but I'd have to look whether they actually tested, did the virus itself generate any other icosanoid storm. That I'm not sure.
No, they didn't. Yeah, I mean, I would say viruses are pretty good at activating prostaglandin biosynthesis in all in vitro settings, but in vivo, I think the real answer here is, as Darryl pointed out, the temporal analysis. It's really critical here, not only for lipid mediator class switching, but it's very important, I think, in my opinion here, is that, you know, many culture mediums are deficient in omega-3, EPA, and DHA. I mean, they're just left out of the media. So the pathways are skewed towards what the cow ate, like linolenic acid. So we see a lot of this in a lot of papers. You know, people are they don't realize it's coming from the cow. And so there are very clearly organs in the human body that sequester DHA and EPA, even when there are low levels. And those might be target tissues where some of the lipid mediators are maybe more critical than others. And we need to perhaps think about that. And the pool that's supplemented might not be the same precursor pool for production and action. This we have to acknowledge as well. But as you know, my own personal approach is to try to mimic nature and make mimetics and analogs that will do what the resolvins do. And that's been a quest that's ongoing. So it takes a long time, but one other area that I think is really emerged that could be a target site for long haul COVID are the GPCR receptors that we've identified for the resolvins themselves. I mean, they're wide open to making mimetics. There was one very exciting paper came out recently from a group in France showing an agonist antibody to the resolvin E1 receptor stimulated resolution and activated the production of other resolvins in vivo. So there are multiple approaches for the long haulers, which I think we're going to see around for a long time. So there might be research opportunities at each one of the levels here. So I guess we can ask each other questions because I don't see any other questions coming in from, and there's no way to tell from the response monitor if those that have asked the question are satisfied with the answer, unlike a live meeting. So,
So
you know,
Charlie, I've got one for our colleagues that follows up on what you were talking about a little bit.
okay.
You know, we talked about nutritionally, you know, trying to increase the levels of the SPMs. And Darryl, I really liked your approach using a drug that's been approved, at least using phase 1 studies. But what's, you know, Dipak, Bruce, and Charlie as well, you commented on some. What's the state of the art for pharmacology of SPMs and approaches to increase them?
Well, I can say in the cancer world, we take, like, for example, aspirin is triggers the SPMs, but aspirin itself has some toxic effects. But the cancer preventative studies, the Lancet papers with Rothwell and stuff, there's definitely some intriguing data about aspirin and anti-cancer activity and stimulating resolution. We had a JCI publication about 2 years ago. We showed a drug that's used for pain called Toradol, ketorolac, that can stimulate SPMs. And if we gave it right before surgery, you could stimulate resolution and prevent some of that surgery stimulated tumor growth we showed through lipoxin and resolvins. So we are definitely interested in drugs that are normally given, for example, in cancer patients. But the problem has been toxicity. Like Toradol has bleeding issues. The surgeons in the clinical trials were very scared about giving cancer patients right before surgery. They're okay giving it after surgery. Same thing with aspirin. Giving it before surgery was, you know, most people come off the aspirin. But that's something we're actively looking at, other ways to boost the resolution. In mice, we gave a high omega-3 fatty acid, aspirin, and resolvins, RBD4, RBD5, or an SCH inhibitor, and we could boost resolution right before we did the surgery in animals.
All right, just let me jump in and answer Rich's question without trying not to be too coy. So, you know, the big barrier, as simple as the SPM molecules are, they're really difficult to synthesize. 22 steps. Sometimes it takes us 5 years to establish the synthetic route and match to the endogenous product. You know, now today we have a biochemical pathway, but all the nuts and bolts that went into it, sometimes, you know, people don't want to dig that much into the chemistry. So that was a major problem in scaling up for GMP synthesis of a resolvent for therapeutic analysis. And that was really disappointing to me personally, to recognize that the cost of making the potential drug outweighed people's value of the benefit. Okay, so that being said, that's no longer a problem. The synthetic routes to many of the resolvents have been shortened significantly, and there's been fine work from a number of synthetic organic chemistry groups that have achieved that. That being said, NIH was very gracious and awarded our P50 grant money to do a phase 1, phase 2 trial in oral inflammation and periodontal disease, where we designed a mouth rinse. And I can tell you that that was a positive phase 1, phase 2. It has some pretty striking results. And I'm not on the manuscript. That's what just recently submitted. Tom Van Dyke's group submitted that. And I just want to bait you to take a look at it as soon as it comes out, because one of these principles that have been really fascinating here and that I think can work for us. And when we think about, I haven't seen this in any other biochemical system with autocoids, that a very small amount of a resolvent, once it's activated, stimulates a feed-forward domino effect in producing other resolvents and amplifying the pro-resolving response. And I think that helps us a great deal on the pharmacology and the therapeutic side. So yeah, do you want to add to that in some of your studies, Dipak, on resolving E1 that you're getting, ready for human?
Yeah, it's actually excited because in our cancer models, we were giving via mini osmotic pump or giving it IP. But for clinical translation cancer patients, we have with Fetus an analog of resolving E1 that we gave orally versus subcutaneously. And the most potent activity we actually got sub-Q. So we're working on a sub-Q delivery system, and we only had to give it once a day. And we've basically, we're looking to test in pancreatic cancer, colorectal cancer, maybe in brain cancer. We have a manuscript also in revision at JCI about resolvents crossing the blood-brain barrier in brain cancer, and the protectins especially were pretty good in brain cancer. So our Fetus is really, taking some serious steps to put a resolving E1 analog into cancer trials for not this year, but the following year to start.
Great, thanks, Dipak. So there are some additional questions that have come in. The first one is from Harold Akemma. I hope I have that pronounced correctly. And it's to Dr. Zeldin. And he says, thanks for the answer about the SH inhibitor. I was wondering about the 6-keto PGF1 alpha because it seemed like it responded differently to the first data you showed. So wondering about how the SH inhibitor affected it, if it was also opposed to prostaglandin D2 and thromboxane. It's jumping on the screen here, hard for me to read. Effect, it would support the contention that the effect on prostanoids is indirect. I think you answered that already, Darryl. But
so the 6-keto data that I showed was from Benji Schwartz's model without the SEH inhibitor.
yeah.
But let me look and see. I can tell you what in our model, whether I can tell you whether TPPU. Let's see here. I guess I could share my screen. So the effect of TPPU on 6-keto PGF1 alpha, there's no consistent effect. on day 5 or day 7. So compared to vehicle, on day 5, TPPU increased 6-keto PGF1 alpha, and on day 7, there was no change compared to no change. So it's inconsistent, and the error bars are pretty high. And I didn't show that data. So I don't know if that answers the question. But.
Well, it may also be targeting, endothelium at a different, versus. Yes, but I think that addresses that. There's another question looks pretty interesting here from Dr. Liang. How does the body/cell process the excessive amounts of SPMs? Is there another quote unquote resolution process for SPMs themselves? Also, thank you all for wonderful talk and discussion. So yeah, Bruce, do you want to take this one? Because this is the resolution of resolution. You know this.
Well, I think Charlie really should answer this because he outlined the metabolic pathway for inactivation of each of the SPMs and in his elegant work. But suffice it to say, it mirrors what happens to prostanoids and leukotrienes, and there's specific enzymes, 15-PGDH, as well as eicosanoid oxidoreductase, that are pivotal to converting the active molecule into an inactive molecule. But all that work was actually laid down by Charlie, so I feel a little bit out of sorts answering it in his presence.
So Charlie,
but
have you thought about maybe targeting those degrading enzymes as a way to increase endogenous levels of the pro-resolving compounds in an acute setting, like just knock off similar to the SEH inhibitor story, knock off the enzymes that degrade it?
I confess we've been slaving away at it, for a long time. But yes, I think that's a very important direction to go in. But I think the biology here is really cool. It's telling us that there's a place in time for each process, and it has to go on properly and it has to come off properly. And it's like one whack is not going to be beneficial in the future. So we really need to know about when and where is the right timing to give something. And but absolutely, I mean, the first round through this was to make mimetics that would resist rapid inactivation. And that followed some of the early, prostaglandin stable analogs, which was so successful as drugs. But now we're moving along those lines in more detail to try and prevent local inactivation. Luckily, we have some target compounds out there already. So I think we're close to closing time, and I have to say I think it was a fantastic session, and I hope that all those members of the American Oil Chemists Society got as big a kick out of this as I, because it really brings the lipids to the center stage, which is where they truly belong in this disease setting. And I'm going to go out on the limb and say, because we're all lipid lovers here, that the reason why there's so much data on the cytokine storm has to do with the ease in which they can be measured, and they can be measured by ELISA assays and by multiplex assays very easily. And when we get all the lipid mediators together in a platform like that, which we cannot at this time because they all have even though they look chemically similar on paper, their physical structures and chemical properties are so different, they cannot be put on all the same platform. It's even hard to get them out of one gradient system. But when we get to that level, then the lipid mediators will again be center stage, right? As they are in this process. So I want to thank the speakers. If you have any closing remarks, now's the time to say it. So I have to thank Dr. Levy. I have to thank Dr. Panigrahy. I have to thank Darryl Zeldin for a home run. And I have to thank Richard, and I have to thank Philip also. Do you guys have any closing remarks, pearls of wisdom for the audience?
Can I just thank you, Charlie, and thank Richard, thank Philip, especially Philip, for staying up to midnight probably, and to my colleagues Dipak and Darryl, and to the audience for staying up with us. It was really an honor to be part of the session.
Great symposium.
Likewise.
Yes.
Yeah,
Great.
great session. Thanks very much, everybody.
Thank you. And the recording will be available in, I think they said 48 hours or so, and then we'll get the recording out to you guys as well. It'd be fun to have as in your diary pack, right? Thanks. I'll write to Patrick tomorrow about that. But thanks so much. Thanks, everybody.
Thank you.
Ciao.
Thank you, everybody.
Good evening. Bye.